So good morning everybody. Today we are going to discuss about the second chapter of hatchery management. So there are so many factors we need to follow in the development of hatchery, the management of hatchery. So in addition to that, today we are going to discuss about the development of the sexual products. So how do we develop the sexual products in the hatchery? So development of eggs. The sexual products are nothing but eggs and sperms. So how do we develop the eggs in the hatchery? So stage one. In stage one, the primitive egg cells are ovogonium or archegonium are very small, their size being hardly bigger than that of other cells. That is 8 to 12 microns. They multiply by normal mitosis. So development of eggs. In the first step of development of the sexual products is that the eggs. The primitive egg cells are very small in size. They need to multiply by normal mitosis. In the second step, the egg cells grow to a size of 12 to 20 microns. In the initial stage, the size of the egg is about 8 to 12 microns. In the second stage, the egg cell grows up to a size of 12 to 20 microns and a follicle begins forming around each egg cell. The follicle, whose function is to nurture and protect the developing egg, eventually becomes a double layer of cells. So, after reaching a size of 12 to 20 microns, the follicle formation starts around the egg cell. The follicle function is to nurture and protect the developing egg. Eventually becomes a double layer of cells. So, it can provide double layer of cells for protecting and nurturing the egg cell. Now coming to the third stage. What happens in third stage? During this stage, the egg cell grows significantly larger to attain a size of 40 to 200 microns and becomes enclosed by the follicle. So the larger size, when the egg attains a larger size of 40 to 200 microns and it becomes enclosed by the and it is completely enclosed by the follicle. These first three stages mark the period prior to the accumulation of nutrients in the developing eggs. So these first, second and third stages are mark the period prior to the accumulation of nutrients in the developing eggs. Now coming to fourth stage. During this stage, the production and accumulation of the yolk begins. This is the process known as vitellogenesis. So what happens in the fourth stage? The production and accumulation of the yolk begins. This is the process known as vitellogenesis. The yolk production takes place. The process is called vitellogenesis. The egg continues to grow to a size of 200 to 350 microns with the accumulation of drops of lipoid materials in its cytoplasm. So during this stage of vitellogenesis, the egg continues to grow to a size of 200 to 350 microns with the accumulation of drops of lipoid materials in its cytoplasm. In its cytoplasm, the lipid molecules develops. Now coming to the fifth stage. What happens in the fifth stage? This marks the second phase of vitellogenesis. So fifth stage marks the second phase of vitellogenesis. The cytoplasm is now full of lipoid drops and yolk production begins. The egg size reaches 350 to 500 microns. So during fifth stage, second phase of vitellogenesis takes place. What happens in the second phase? The cytoplasm is now full of lipoid drops and yolk production begins. So the full cytoplasm is fully developed with the lipid molecules and yolk production begins. The egg size reaches 350 to 500 microns. Now the egg size reaches to 500 microns. Now coming to the sixth stage. What happens in the sixth stage of egg development? This is the third phase of vitellogenesis. During this stage, the yolk plates push the lipoid drops towards the edge of the cell. So during this phase, what happens? The yolk plates push the lipoid drops towards the edge of the cell, where two rings begin forming. So at the edge of the cell, where two rings begin forming, the nuclei which take part in protein synthesis and the accumulation of nutrients are seen ordering to the membrane of the nucleus. So the size of the egg is now 600 to 900 microns. Now the size of the egg reached to 900 microns at this sixth stage of vitellogenesis. So what happens in the sixth stage of egg development? This is the third phase of vitellogenesis. During this phase, the yolk plates push the liquid materials towards the edge of the cell membrane, where two rings starts forming the nuclei. One of them is forming the nuclei which take part in protein synthesis, and the accumulation of nutrients are seen adhering to the membrane of the nucleus. So the size of the egg is now become 900 microns. So what happens in the sixth stage actually during the third phase of vitellogenesis, the lipoid drops are pushed towards the edges of the cell where two rings begin to form. Then the nuclei take part in protein synthesis and the accumulation of nutrients are seen adhering to the membrane of the nucleus. So the size of the egg now become 900 microns. So these are the changes occur during this sixth stage of egg development. And now coming to seventh stage. What happens in the seventh stage of egg development? The process of vitellogenesis is completed during this stage. So the vitellogenesis process is completed during seventh stage and the egg attains a size of 900 to 1000 microns. So the finally egg attains 1000 microns. When the yolk accumulation ends, the nuclei withdraw into the center of the nucleus. When the yolk accumulation ends, the nuclei withdraw into the center of the nucleus. The nucleoli withdraw into the center of the nucleus. The micropyle, a small opening on the egg shell, develops during this stage. So during the seventh stage of egg development, what happens? The accumulation of yolk stops are closed. Then what happens? The size of the egg attains 900 to 1000 microns. And when the yolk accumulation ends, the nucleoli withdraw into the center of the nucleus. The micropyle develops during this stage. The micropyle is a small opening on the egg shell that can be developed during this seventh stage of egg development. So stages 4th, 5th, 6th and 7th are the stages of vitellogenesis. So what do you say here? The stages 4th, 5th, 6th and 7th are the stages of vitellogenesis. When yolk is synthesized and accumulated in the egg cell, the egg is now materially ready. So when these 4 stages were completed, when the yolk is synthesized and accumulated in the egg cell, the egg is now materially ready. To reach this stage of development, the female fish needs a lot of protein in its food and a favorable temperature range. So to reach this stage, the egg is reached to this stage of development. The female fish needs a lot of protein rich food. So the female fish needs a protein rich food and favorable temperature conditions for the developing of egg. Now further development of the eggs towards ovulation or final ripening is regulated by gonadotrophin hormones. Further, what happens that the development of the eggs towards ovulation or final ripening is regulated by gonadotrophin hormones, which are formed and stored in the pituitary gland or hypophysis. So, the further ovulation is regulated by gonadotrophin hormones. So, these are sexual hormones which are formed and stored in the pituitary glands or hypophysis. Some hormones of the hypophysis, such as FSH, follicle stimulating hormone, and LH, luteinizing hormone, are continuously produced and secreted into the bloodstream. So, what happens? The gonadotrophin hormones are the reproductive regulatory hormones. So, the gonadotropic hormones, which are formed and stored in the pituitary gland or hypophysis, the hormones include the follicle stimulating hormone, that is FSH, and luteinizing hormone, that is LH, are continuously produced and secreted into the bloodstream. These two hormones are directly secreted into the bloodstream, continuously secreted into the bloodstream to regulate the ovulation process. At the reproductive age. Now coming to, on the other hand, the steroid type of hormones like estrogens secreted by the capsule, that is theca, of the follicle inform the brain about the stage of egg development. So on the other hand, what happens? The steroid type of hormones also regulate the egg development, the stages of egg development. So on the other hand, the steroid type of hormones like estrogen secreted by the capsule, that is theca, of the follicle inform the brain about the stage of egg development. So these estrogens can inform the brain about the stages of the egg development. On the completion of stage 7th, the egg may remain as such for several months without any change, and this forms the dormant or resting phase. So on the completion of stage 7th, on the completion of the development of the egg, the egg may remain as such for several months. After completion of the 7th stage of development, the egg remain same for several months without any change, and this forms the dormant or resting phase. So this resting or dormant phase will either end in ovulation if favorable conditions occur, or in follicular putrefaction, and resorption in the absence of such conditions. So what happens? After completion of the 7th stage of development, the egg will remain same without any change, and this type of form is called dormant or resting phase. During this resting phase or dormant phase, uh, this resting or dormant phase will either end in ovulation if favorable conditions occur. This resting phase end with ovulation if the conditions favors, or in follicular putrefaction and resorption in the absence of such conditions. If the conditions are not favorable, these eggs can be putrefied or resorption means reabsorbed into the follicle in unfavorable conditions. If the conditions are favorable, the egg can be ovulated. Okay. So these are the conditions or methods which are involved in the egg development and ovulation process. And when the environment changes for better, the fish starts gathering through its sense organs all the necessary information about environmental conditions, example temperature, suitable place for spanning, water current floods, water current, floods, presence of fish of the opposite sex, etc. So when the environment changes for the better, so whenever the environmental conditions are optimal for the fish production, the fish starts gathering through its sense organs all the necessary information about environmental conditions. uh, Example, temperature, suitable place for spanning, water current, floods, presence of fish of the opposite sex. So all these things can be considered for the development of the fish or for the reproduction of the fish. So fish needs to optimal conditions for releasing eggs and also sperms for the further development. So what are the conditions they need actually? When the environmental changes for better optimal conditions, when the environment changes for the better, the fish starts gathering through its sense organs. So the fish gathering through its sense organs can smell the optimal conditions in the water. So all the necessary information about environmental conditions like temperature, suitable place for spanning, water current, floods, the presence of fish of the opposite sex, all these things are available at that place means the fish will gathered and they will release eggs and sperms for the further development. So all these things are necessary for the fish to develop. And this sensory information accumulates in the hypothalamus of the brain. The sensory information is accumulates in the hypothalamus of the brain, and when a certain threshold level is reached, the hypothalamus gives an order through a hormone, that is gonadotrophin releasing hormone GRH, to the hypophysis to release gonadotrophins into the bloodstream. The gonadotrophins thus released reach the gonad and trigger the pre-ovulation process and final ovulation. See how the process, how the mechanism takes place uh, during ovulation or before ovulation. So the sensory information accumulates in the hypothalamus and brain, and when a certain threshold level is reached, the hypothalamus gives an order through a hormone, gonadotropic releasing hormone, to the hypophysis to release gonadotrophins into the bloodstream. The gonadotrophins thus released reach the gonad and trigger the reach the gonad. When the gonadotrophins reach the gonads and trigger the pre-ovulation process and final ovulation. So this is all about the ovulation process. The fish effect of gonadotrophins on the egg. The first effect of gonadotrophins on the egg is the movement of its nucleus towards the micropyle. The first effect of gonadotrophins on egg is that the movement of its nucleus towards the micropyle. This is followed by what is termed as hydration, wherein the eggs absorb water. All these are completed during the pre-ovulation stage. So what happens in the first stage of gonadotrophins reach the gonads means egg is gone. What happens? The egg is the movement of its nucleus towards the micropyle takes place. Within the egg, the movement of its nucleus towards the micropyle takes place in the first step of gonadotrophin on the egg. This is followed by what is termed as hydration. This is followed by hydration. Hydration takes place. Means water absorption takes place, wherein the eggs absorb water. All these are completed during the pre-ovulation stage. The hydration and nucleus movement towards the micropyle and hydration takes place. Um, all these things completed during the pre-ovulation stage. Now coming to after pre-ovulation, what happens? The membrane of the nucleus disappears. The chromosomes become visible. After pre-ovulation, what happens? The membrane of the nucleus disappears, the chromosomes become visible, and the first cell division of meiosis occurs, during which the total number of chromosomes is reduced to half. At the same time, the follicle keeping the egg fixed onto the wall of the ovary. At the same time, what happens? The follicle keeping the egg fixed onto the wall of the ovary gets dissolved by enzymes, and the ripe for fertilization egg falls into the cavity of the ovary. So, after pre-ovulation completes, the membrane of the nucleus disappears. So, the nuclear membrane disappears, chromosomes become visible, and the first cell division takes place, that is meiosis. Means the chromosome number will be reduced to half. At the same time, the follicle keeping the egg fixed onto the wall of the ovary. At the same time, what happens during that meiosis? The follicle keeping the egg fixed onto the wall of the ovary gets dissolved by enzymes, and the ripe for fertilization. That egg gets ovary gets dissolved by enzymes, and the ripe for fertilization. Now the egg is ready for fertilization. Egg falls into the cavity of the ovary. Now that egg enter into the cavity of the ovary. The second cell division of meiosis normally takes place in the presence of the sperm. The second cell division of meiosis normally takes place in the presence of the sperm, which intrudes into the nucleus of the egg, through the micropyle. So the sperm enters into the egg through the micropyle during the second meiotic division. Therefore, the presence of the male pronucleus is necessary for the development of the female pronucleus. So, what we need to understand here is that the presence of the male pronucleus is necessary for the development of the female pronucleus. Now coming to oogonial proliferation phase. How the proliferation takes place. First, vitellogenesis phase, and then the resting or dormant phase takes place. First, vitellogenesis takes place during the egg development. During egg development, the vitellogenesis takes place. After vitellogenesis, resting or dormant phase comes. After resting phase, the egg will ready for ovulation or spanning, or in favorable conditions, the egg will go to ovulation or spanning. If the conditions are not favorable, that can be that can go to resorption or putrefaction. So reabsorbed by the follicle. That is nothing but reabsorbed by follicle. Now coming to what happens during ovulation. What happens during ovulation means migration of the nucleus to the periphery of the egg near the micropyle. During ovulation, the steps. What are the steps involved or take part during ovulation? Means migration of the nucleus takes place to the periphery of the egg near the micropyle. A second, hydration takes place, means absorption of water by the micropyle into the some membrane. Okay. So next, hydration takes place, that is absorption of water through the micropyle into the egg. And next, breaking or rupture of the follicle can be done. When the egg cell takes the water, that can be bulged, that area can be bulged and then ready to break down or rupture of the follicle. And first phase of meiosis, The fourth step is that first phase of meiosis, cell division of the meiosis, in which the number of chromosomes is reduced to one half. So what happens during ovulation means, during ovulation, first the migration of nucleus takes place towards the periphery of the egg near the micropyle. The hydration takes place means the absorption of water can be done. Now breaking or rupture of the follicle takes place. Again, the first phase of meiosis can be done. In the development of sperm, what happens during the development of sperm? The process of development of sperm is far less complicated than that of eggs. So the process of sperm development is less complicated than the egg. So you can see the flowchart of spermatogenesis here. Spermatogonia, propagation by mitosis. Spermatogonia undergo mitosis and development of primary spermatocytes and later two secondary spermatocytes form. Then production of two spermatozoa or sperm in each spermatocyte can be done. Then collection of sperm in the cavities of the tubules of the testis in a dormant state. So characteristics of the sperm. What are the characteristic features of the sperm? The sperm are motionless. Sperm are motionless in testis. The sperm are motionless in the testis but become motile when they come in contact with the water. So the characteristic features of sperms are they are motionless in the testis. Sperms are motionless in the testis but become motile when they come to contact with the water. The motile period of the sperm is very short and depends on the temperature of the water. So the motile period of the sperm is very short and depends upon the temperature of the water. Okay, now coming to the other characteristic feature of sperm is that the sperm of warm water fishes move actively by their filamentous tail for only about half or 1 minute. The sperm of warm water fishes move actively by their filamentous tail. The sperms will move actively by their filamentous tail in warm water fishes. If the sperm belongs to warm water fish that can move actively by their filamentous tail for a minute or half minute. The fish sperm are very small. Their estimated number in 1 cubic centimeter of milk being about 10 to 20,000 million, depending on the denseness of the milk. So the fish sperm are very small. They are very small in size. Their estimated number in 1 cubic centimeter of milk being about 10,000 to 20,000 million, depending on the denseness of the milk. So one milk will contain 10,000 to 20,000 million sperms. It is depending upon the denseness of the milk. So how much dense it is, the number of sperms can be increased. So it's depending on the denseness. It is more dense, the number of sperms will be more. If less dense, the number of sperms will be less. Now coming to fertilization and fertilized egg. Fertilization and fertilized egg. The eggs that have fallen into the cavity of the ovary do not have definite shape. When the eggs are entered into the cavity of the ovary, do not have a definite shape. The egg shell is soft and tightly surrounds the cell kernel. which includes the nucleus and the mass of the yolk. So when eggs enters into the cavity of the ovary, they are not having any definite shape, and also they have soft and tightly surrounds the cell kernel, which includes the nucleus and mass of yolk. So egg contains nucleus and mass of yolk with a soft and tightly surrounded kernel, and it will not have a definite shape. Okay, during fertilization or after fertilization. Now coming to when the ripe egg falls into the water, when the ripe egg falls into the water, it assumes a round shape and within a short time begins to swell. See how the difference takes place in the water. When the egg, when the ripened egg falls into the water, it assumes a round shape. In the cavity of ovary, there is no shape for the egg, but when it ripened and released into the water, it will get a round shape and within a short time begins to swell. The water penetrates between the shell and the kernel of the egg, and thus the perivitelline space develops. So that the perivitelline space develops by the absorption of water. The water penetrates between the shell and kernel of the egg, and thus the perivitelline space develops. Now coming to the next step, the micropyle closes within a minute. After absorption of water, the micropyle closes within a minute, after which no sperm can enter the egg. When the micropyle closes on the egg, the sperms cannot be enter into the egg. The swelling usually lasts for about 1 to 2 hours. The swelling of the egg may be lost within 1 or 2 hours. The egg then takes its final form and becomes water hardened. The egg then takes its final form and becomes water hardened. The swelling, however, does not alter the size of the kernel. So the swelling occurs, but that does not alter the size of the kernel. So these are the things can be happen after releasing the egg into the water. Now coming to fertilized fish eggs can be of different types. So now what happens? First the eggs, the formation of eggs can be done. The ovulation takes place. After ovulation, that can be enter into the cavity of the ovary. At the time, the egg will not have any definite shape, and after released into the after ripening, the eggs released into the water. When they enter into the water, they will get a definite shape, and micropyle will be closed so that the sperms cannot be enter into that egg. So all these steps involved in the involved during the fertilization or egg development process. The fertilized fish eggs can be of different types. The fertilized eggs can be of different types. However, for practical purposes, two principal categories can be distinguished. So there are different types of fertilized eggs, but practical purpose there are two categories are distinguished. They are non-adhesive and adhesive. They are of two types, non-adhesive and adhesive. Non-adhesive eggs means non-adhesive eggs again are of four types. Include buoyant eggs, floating eggs, semi-floating eggs, and rolling eggs. Buoyant eggs means specific weight slightly less than that of water. So buoyant eggs are nothing but they have specific weight slightly less than that of water. Uh, floating eggs are floating eggs also have a specific weight that is slightly higher than that of water. So buoyant eggs they have specific weight but slightly less than the water weight. Floating eggs also have a specific weight, but that's slightly higher than that of water. And semi-floating eggs are specific weight. They also have a specific weight that is higher than that of water. Rolling eggs have specific weight like other eggs. It is higher than that of water. So these are the differences between these four types of eggs. Now coming to what determines the specific weight of eggs. What determines the specific weight of eggs? The specific weight of eggs is determined by the size of the perivitelline space. The specific weight can be determined by the size of the perivitelline space. If the perivitelline space is large, the egg size will be increased. If the perivitelline space is decreased or the size is less, then the egg size is less. The specific weight of the kernel affected by presence or absence of oil droplets. So the specific weight of the kernel affected by presence or absence of the oil droplets. Now coming to adhesive eggs. So these eggs have an adhesive layer on their shell. These eggs have an adhesive layer on their shell. These becomes activated when the egg comes into contact with water. Adhesive eggs are having adhesive layer on their shell which becomes activated when the egg comes into contact with the water. The adhesive layer sticks the eggs to some objects or to each other. So this adhesive layer sticks the eggs to the objects or to each other. So this is another type of eggs. One is non-adhesive, second is adhesive. Non-adhesive, they don't adhere to any object or adhere to each other. But in adhesive eggs, they are having adhesive layer and attached to the objects, attached to some objects or to each other, eggs to each other. So these are the two differences between the eggs. And now coming to two types of attachment can be distinguished. Eggs sticking to objects, eggs sticking only to each other, forming a clump or egg mass. So two types of attachment can be distinguished. See, before we have discussed about the adhesive and non-adhesive eggs. Now we are in adhesive type. The eggs can be distinguished in two types. One is that eggs sticking to objects, and the second is that eggs sticking only to each other, forming a clump or egg mass. So again, two types. Adhesive nature can be distinguished into two types. One is to objects, other is to eggs attached to each other. Now coming to the adhesiveness can either be strong and the eggs suffer damage, it turn off from the substratum. Second, quite weak and eggs can be easily removed. So if the adhesive object, if the adhesiveness is strong enough, means the egg cannot be easily released from the substratum. It can be damaged while leaving the substratum. If the adhesiveness is quite weak, then the eggs can be easily removed from the substratum. So this is another factor which is related to adhesiveness. And next, variations. What are the variations in adhesiveness? The adhesiveness gradually weakens during the course of egg development. So the adhesiveness is gradually weakens during the course of egg development. Higher salinity affects the negative influence and adhesiveness. If the water contains high salinity, that affects the negative influence and adhesiveness. The fish eggs vary in size. The eggs of fish can be vary in size. Now coming to the factors which determine the size of the egg are the factors which determine the size of the egg are the size of the egg kernel, the thickness of the eggshell, size of the perivitelline space. These three things can determine the size of the egg. The size of the egg kernel, the thickness of the eggshell, and size of the perivitelline space can determine the size of the egg. The first two determine the size of the dry eggs. The first two things that kernel and eggshell can determine the dry eggs, can determine the size of the dry eggs. That is before they come in contact with water. Dry eggs means before they come to contact with the water. While the third determines the size of the water hardened eggs. While the third determines means perivitelline space that determines the size of the water hardened eggs. Thank you. Thank you for your patient listening. And I thank the source PPTs for sharing their slides for my lesson preparation. Thank you.